Okay, thank you very much, Igor. It's been a pleasure to work with him and his team and many others at JGI. And thank you all for sticking around right before lunch. And I do want to put in a shameless plug. I am hiring. If you yourself or anyone you know of is interested in to relocate to beautiful Santa Barbara, you can see where our lab is, right? Do please keep me in the know. What I will also say, a fun fact about myself is that I have 3 children at or under the age of 5, which brings me to my next slide. We'll see if it goes. Yeah, all right. So poop, it's kind of a big deal. And I'm not just saying this as a bad start to a TED Talk, I promise. I think about poop in some other ways, but as an engineer, I don't mean to gross you out again before lunch. I think of poop as a biotech resource, right? It sounds strange. What do I mean by that? What do we find in poop? Well, we find a lot of diversity in microbial taxa that reflects all of the diverse action that is happening in the digestive tract of ourselves. But what I'm going to talk to you about today is what's happening in the digestive tract of large herbivores. Think cows, think goats, think sheep, things like that. Why? Because they take in crude biomass, like the cellulose, and they get things out of it that we as humans cannot with our digestive tract, right? So if you peer inside of a poop sample or a rumen sample, you're going to find beautiful diversity, and I'm not even going to begin to describe it here, right? But as an engineer, I like to look at this as an opportunity to build communities and teams through synthetic biology and systems biology approaches that can do wonderful things that none of these microorganisms can do on their own. So just to kind of orient ourselves, I love microbial communities because they divide and conquer difficult steps and tasks. The lignocellulose degradation is a wonderful example that I'll tell you about how some fungi and some bacteria work together to do just that. And what's more, microbes are kind of like people. They have their friends, they have their enemies, and they have their frenemies. That social dynamic really does change based on the environment that these microorganisms are presented with, right? This is part of the reason why microbial communities are so stable in the face of perturbation, because they have the ability to change and bounce back, right? Now, my group is really interested in answering questions that sound real basic on the surface. Specifically, who is there? Just what's the membership profile look like? And the harder question to answer, what are they doing, right? And I motivate the talk to say that here's kind of where we're going with that knowledge. One aspect is, how can we learn from nature to essentially take microbes off a shelf and put them together in new and interesting ways to get whatever we want on demand from waste, right? That's a big ask. It really is. It's very hard. We've not figured out how to do it. Others are working on this. This is a long goal. But I would also advocate that there's another, maybe easier path, which is to harness parts from microorganisms. And when I say parts, I mean enzymes, I mean transporters, I mean maybe the microbe itself as a novel chassis system. And that's an easier goal, right? So I'm going to tell you about both of these. And so the objectives of the talk today and the time that we share together is I want to tell you about 3 stories. One is where we're enriching and engineering microbial communities to try and tackle that first big goal of making something valuable using microbial communities that we formulated and inspired from nature. And I'm specifically going to tell you about volatile fatty acid production as a result of lignocellulose deconstruction. Then I'm going to tell you about addressing that, what I would say is an easier ask, right? Trying to find these parts from nature that do wonderful things. So mining rare members of microbial communities and really taking their enzymes, their transporters, their biosynthetic pathways, their novel abilities, and porting them into other systems that we can control. And then with the time that we have left, we'll see how fast I talk. I'll tell you about something that I don't often talk about, which is bioprospecting microbial communities in the ocean off the coast of Santa Barbara to look at plastic degradation. And so to hop right in, let's start with the donors of our poop in our lab most of the time, and that is large herbivores. And why I want to start in that location is because this is a beautifully diverse environment, as you see there, and these are broad estimates. People will fight with me about what this is. I don't really want to fight with people, but as you can see, bacteria vastly overpopulate the system, as they do by the numbers in most other communities. What I'm highlighting there are fungi that you may or may not know exist in the guts of herbivores, particularly obligate anaerobic fungi that Igor was talking about. And I don't have a slide on this, but they are unfortunately discovered and named by a lot of Welshmen. So their naming convention has no vowels and it is really difficult. So don't blame me for it. I didn't do it. But I want to talk about these fungi a lot because they are actually big, heavy lifters. And even though in this community where they exist in low numbers, right? And part of what we're after here is to preserve this beautiful diversity and complexity of microbiomes like what we find in the cow rumen, so that we can actually achieve some of these cool outcomes that only microbial communities can obtain. So I just want to highlight that we want to preserve this diversity. We don't want to just cultivate bacteria, right? And so in an effort that was led by Nick Peng, who's a former postdoc from the group, is now leading his own group at the University of South Carolina, we endeavored down this goal by going to our local Santa Barbara Zoo, finding the happy donor herbivore of that day, whose name is Elway the goat, pictured to the left. Elway was our fecal pellet donor and freshly donated sample. And what we did is we basically took that sample back to the lab and we started to treat that sample in different ways to sculpt and influence the community membership that would result. And our hypothesis was that by growing on 4 different substrates or food sources for the microbial communities, we get different teams to form, right? Not that risky of a hypothesis, I thought. Alfalfa, bagasse, reed canary grass, lignocellulosic substrates with varied degree of pectin and other carbohydrates. Xylan is the most different from the bunch because there's no lignin or cellulose in it. We would expect a vastly different community from Xylan. What we also did is we enrich in triplicate samples over 10 different generations, and we provide 3 different chemical treatments to those teams as they form. One on the bottom is a treatment with chloramphenicol that is there to knock down anything or knock out anything prokaryotic. So we expect to just have a mixture of eukaryotic fungi or yeast that result. The middle treatment is a cocktail treatment of the antibiotics penicillin and streptomycin, which should knock down but not completely eliminate the prokaryotic members of the community, but it was specifically targeted to preserve the interaction of fungi and archaeal methanogens. And then that first treatment is really no treatment at all, just let the microbes fight it out. And as these generations evolve on the different substrates and different members drop out and take over, we are also tracking real-time metabolite analyses through HPLC and GC. And then we're also smashing and grabbing the DNA and the RNA to give us a functional metagenomic window into what's going on. This was a big experiment. And so we were very happy to partner with JGI through JGI CSP to do a lot of the metagenomic aspects of this work. Essentially, you see the pipeline there, which we extracted all of the hundreds of samples, sent them to JGI, did marker gene analysis, but also did full metagenome assembly. And this was key to understanding and answering, in part, that question of who's there and what are they doing, right? We want to assess out metabolic potential. And part of what we found from Elway the goat is, and that is the family tree of microorganisms, similar to what I introduced in the beginning of the talk. We counted about 200,000 carbohydrate active enzymes or Kzymes just from Elway the goat, which is fantastic. I'm not saying that these are all novel, far from it, but it just highlights for you how much biotech is in food, right? These enzymes are very useful for industrial purposes. We were able to put together a vast amount, hundreds, just over 700 prokaryotic metagenomes, and we were able to partially reconstruct about 18 or so eukaryotic metagenomes. That is still an art form. Assembling fungal genomes through metagenomics is very tough because they are giant genomes and they are highly repetitive, especially these fungi. So the fact that we could even see them was great. Importantly, a very small fraction of these microbes were actually enriched through our cultivation. In other words, a lot dropped out, right? But what we see at the bottom here, and this is a little small, so hopefully you can see it, just by looking at the amplicon sequencing, we see vast shifts in the population and kind of that selection or sculpting of the community on the fungal end on the right versus the prokaryotic end on the left. And something that I'm not going to get into great detail on here is that we found also functional differences in these teams. In particular, surprises, such that the microbial communities dominated by fungi, where archaeomethanogens were present, produced a lot more methane, almost twice as much methane than those that are dominated by anaerobic bacteria. And I have some reasons I could explain that, but anyways, gives us a little bit of a view into how to sculpt these microbial communities that again are taking lignocellulose or some or a component of it and turning that into chemicals, right? Here, methane. You may ask, what did Elway the goat get for his trouble? Elway the goat got to grace the cover of Nature Microbiology. So there he is with his friend that just sort of photobombed on the bottom. Okay, so what can we do with this knowledge to actually sculpt synthetic microbiomes that would not see each other in nature, right? So we don't want to make methane. Methane's bad for a lot of reasons unless you can capture it. We were instead inspired by something else that a cow does. In particular, if you peer inside of this complex anaerobic microbiome I just told you about, actually the vast majority of the carbon flow here is absorbed by the cow and retained as volatile fatty acids, about 75% of it. This is why these herbivores are able to graze and grow big and strong, right? And so we wondered if we could reproduce this process as kind of a one-pot consolidated bioprocess to make volatile fatty acids, specifically medium chain fatty acids. And our strategy for doing this is to take these awesome anaerobic fungi that are rare members of this community, but produce a lot of these carbohydrate active enzymes, and to combine them with the power of what's called chain elongating bacteria, some of which you do find in these systems, but the ones that I'm telling you about are not, to the best of my knowledge, found in ruminants. So the idea here is you see in yellow the metabolic byproducts being given off by the anaerobic fungi on the left. They're associating with biomass, producing cazymes, punching through those systems, and then releasing acetate, ethanol, lactate, formate, and hydrogen. And then on the right, chain elongating bacteria that are taking in many, not all, but many of those metabolic end products of the fungi and upgrading them into hexanoate or butyrate or a volatile fatty acid. And so we asked if we could put these two together and just see if they could grow together and actually make these products. So I'm showing you some preliminary data from Elena Blair, who's here somewhere. I think there she is. And she has a poster about some of this work. And what Elena was doing is just mixing and matching at different ratios and waiting a different amount of time and seeing how these products changed in a fungal monoculture versus a bacterial monoculture versus a true co-culture. And what I will point out is that this is complicated because there's a lot of ways to put these together. But what you note, hopefully, is that if you want to look at the difference between the initial and the time that's passed, and hopefully what you see for all of the co-culture examples is that butyrate comes up and lactate goes down. And this changes a little bit based on the inoculation ratio, the timing of the cultivation, et cetera. But it does prove that this is in fact possible to form the synthetic community. It also inspires this other question of, gosh, how do you do this optimally, right? So all of us in the microbiome field know that there are so many ways to put microbiomes together, inoculation ratios, substrates, how do you influence the community? It is truly endless. And so to spare Elena from having to systematically do every single one of these experiments, what we want to do instead is to ask a robot to do it. And so we're collaborating with Chris Lawson on a new DOE DER project to do just this. We affectionately call it the Synthetic Rumen Project, where we are trying to pair these anaerobic fungi with chain elongating bacteria, and then to use robotics under anaerobic conditions to rapidly screen for optimal consortia that produce volatile fatty acids, in this case, butyrate. And we're using the design, build, test, learn cycle to really understand the failed matches and also benefit from that information so that we can in silico design better experiments that get us closer to the maximal yield of butyrate. And so this is a picture of the robot that Chris has built at the University of Toronto, and we are well underway with this project. So we're really excited to see where this goes. And so with that, I want to switch gears and tell you about answering that easier question of, okay, studying these microbial communities, you've just seen it's really hard to mix and match these guys and inoculate them in the right way to always get what you want as fast as you can and to optimize them. It's perhaps an easier question to focus on mining some of these attractive microorganisms in their genomes to get at some parts that we can port into other systems that we do understand and can optimize. And I'm not going to spend a lot of time on the background here, but if you've heard me talk before, you probably could give this slide for me. I show this a lot. This is about how you actually can take the anaerobic fungi from the herbivores and use old school enrichment techniques to select for them and get them down to isolates. So this is how we were able to actually isolate from Santa Barbara Zoo animals several of the strains that you see here: Pyromyces spinus, Enteromyces robustus, Neocalamastix californiae, and Cecomyces turovis, which are all from the Neocalamastecomyces. And we can grow them in liquid culture anaerobically. They grow on unprotruted biomass. And we were able to work with Igor's team to publish all of these genomes, which are there in the citations. And several years ago, just by studying the transcriptomes, we published a paper in Science that essentially just counted the number of Kzymes that were homologous to other enzymes that we knew. And we found that these isolates were chock full of Kzymes, especially those that we do not yet understand, including complexes or fungal cellulosomes. And so with that kind of set us on this other path of what else can we find now that we've got these fungal genomes. And so one thing we were interested in is just doing a bioinformatics survey of the membrane proteome, the gatekeepers of the cell, that help us to take in different sugars, right? This helps cells take in different sugars and extrude things they don't want out. We were specifically hunting for carbohydrate transporters, things that uptake glucose or xylose or other 5 or 6 carbon sugars, and also transporters that confer tolerance in the face of difficulties, such as through fluoride, drug exporters, things like that. And we found a lot of these carbohydrate transporters. And what's more, we found that we can put them in a useful system and they still work. So for example, we took a sugar transporter that is, in my view, from this is the best named acronym I've come across. This is a multi-pass sugar transporter called a suite. Sugars will eventually be exported transporter, only bested in acronym by the prokaryotic version that is half a 3-TM protein that is called a semi-suite. That's even better. So we took these sweet proteins that we identified from the anaerobic fungal genomes, codon optimized them, and put them in transporter deficient strains of the Saccharomyces cerevisiae. What you see on the plate there is that the transporter deficient strain will not grow without the ability to transport sugar. But when you put in just the transporter from the anaerobic fungi, voila, they are now growing again. So this is the first proof that we know of that you can actually move some of these transporters over and get them to function in yeast. And what's more, we found that this Neocalamastix californiae, or NC suite 1, also afforded pretty much equal rate uptake of xylose and glucose, which is a big deal in the yeast world because yeast traditionally struggle to take in 5 carbon sugars. So it might open the door in some directions there. What's more, we mined the genomes and we found some other surprises. So we found the building blocks for a lot of polyketide synthases and RPSs and natural products, right? So what we're doing here on this bar graph is just very simply plotting the output of anti-smash that's telling us and scanning the genomes and telling us what those building blocks are. If you're not familiar with natural products, I think many of you probably are, but we're interested in these things because they can be building blocks for other molecules, they can be antibiotics, they can be novel materials, et cetera. And so working together with Nancy Keller at Madison and Trent Northern, who's here, there he is, we were able to, with Candace Swift, who's a previous graduate student in my lab, analyze many of these clusters. We found a quarter of them are transcriptionally active, and we used metabolomics capabilities at JGI with Trent's group to really try and identify what some of these natural products are. I'm showing you one that we think these fungi are producing called Bauman. There's unfortunately no standard for it, so this is the best guess, but we did find through molecular networking, GNPS, that there are many structures yet to be determined if we can try and amplify these from the system. And so Laza Bokovic, who's the current grad student in my lab right now, had a poster on this and is carrying this work forward. And what's more, there's also another surprise, it's somewhat controversial, but that is that there might also be, there is an anaerobic lignin breakdown mechanism in these fungi. I am not an expert on 2D NMR, but what I have been told through collaborators at the Joint Bioenergy Institute is that these spectra show the almost complete disappearance of the beta-O4 linkage when fungi have been added to sorghum cell wall samples, as you can see for these 2 isolates here. I'm happy to entertain more about this. I wish I could tell you what the mechanism is. I can tell you it looks nothing like what aerobic fungi do. But just it goes to show you that there's a lot hiding in these weird systems that we want to still understand. And with the last couple of minutes, I want to pivot a little bit and tell you about how we've applied some of these principles to a new problem and looking at how plastics associate with microbes. And we are located in beautiful Santa Barbara. And so this figure is very close to our heart, right? A lot of the plastic that we generate through humanity does end up in the ocean eventually. And this plastosphere, or how microbial communities associate with plastic, is very much not well known. But we in Santa Barbara have a few different interesting microbial sites that we hypothesize might be playing a role in this plastic association when the plastics get themselves through the Santa Barbara Channel. And one of these places is called Bryan's Seep. So you may or may not know that the Santa Barbara Channel is a hotbed for natural oil seeps. So there's a lot of really interesting microbial activities on the seafloor. I'll show you in just a minute. And we thought, in our hypothesis here, is that where these oil seeps are in these microbial communities might be some interesting association and or maybe degradation or change or alteration activities that these microbes might have against man-made plastic. So we chose to look at the microbial population in Bryan's Seep that is releasing and seeping not only crude oil, but methane, which has a lot of sulfate-reducing bacteria. And what's more, hopefully this video will play. If not, it's not super important. This is actually what one of those seeps looks like. You can hopefully see bubbles coming out. Those are actually methane. And so what we did is we wanted to capture the microbial population that's associated with those sites. And how we did it was actually by dropping cages of encased plastic and in dialysis bags and kind of hoping for the best. I will say that we didn't really know what to do. And if you look at the timestamp of what's going on here, it adds insult to injury. So we dumped these, we affixed through approval these cages down next to these oil seeps, and then we hoped that they would capture some of these unique microbial communities that would assimilate onto the plastics, and then we could use sequencing tools to figure out what colonized these plastics. But as you can see, we kind of go with our scientific dive team, make sure that it's still there, not washed away. And then right after the lockdowns happened, our scientific dive team found that our plastic was destroyed not by microbes, but by the action of fish that were laying eggs in there. So fish literally ate our experiment. And so we had to rethink how to do this. So we pivoted to an ex situ experimental design where we essentially captured the sediment at those sites I showed you, brought them back to the lab. We have a seawater facility at UCSB. And so we kept the fish out, kept the microbes in. And what we did on several different forms of plastic that you see, and I won't get into all the differences, is that we are enriching different taxa of here, just of bacteria and prokaryotes relative to what's in the sediment, relative to what's in the seawater. And this is literally hot off the presses data, but you do see large differences between cellulose acetate, which we use as a control, and all of the other plastic samples, which are on the left, and all of the samples and glass as a control. And so we're still trying to figure out what this means. But I do think that these types of opportunities are, there's a lot that we can still push for to understand what's colonizing these plastics. And so to wrap up, I hope that I've shown you that natural communities are wonderful to study and to inspire us to build synthetic communities. And sculpting those communities is really a hard problem. There's a lot that we and others have to do to tackle that. But there's opportunities for borrowing and porting these parts from nature, enzymes, transporters, pathways into microbes that we do know to advance our biotech goals. And because I'm an engineer, I always love to end on this note. I think we need more engineers out doing bioprospecting, out in the field, doing these types of things. Not only because engineers tend to be a little naive and we think that we can kind of solve these problems, but I think we offer a new perspective. And there's always a better chassis organism to start from. So I'm all for embracing non-model microbes. And I hope that I've inspired you all to do that too. And with that, I will thank JGI in particular through the FICUS and CSP programs, and Igor's group especially, for all of the wonderful help that's really enabled our science. And I particularly want to thank all of my group, which is half of them are here. Hopefully you met them or you will. They're not all pictured in this lovely picture from my backyard, but they're all the folks who are boots on the ground doing the work, and they are tremendous. So with that, thanks for listening, and I am happy to take any additional questions.
No one else asked. Thanks a lot for a very interesting talk, Michelle. Can you provide some context on why you chose a natural gas site as an enrichment experiment? Because the enzymes that are involved in the degradation of those gases are probably not the ones, right, that attack the plastics, because it's a gas versus a solid. And like, can you just, why did you choose
so
that sample?
So there's seeps there, and I probably should have gone over this more. There's also there's crude oil coming up too. So if you actually walk the beach of Santa Barbara, my group knows this and probably done it, you have to bring like olive oil with you or some other wipe for your feet because you will get crude oil and tar stuck to you. So that's the activity we're looking for. My material scientist friends tell me that the structure is very similar to some of the plastics that we've studied. And, you know, I'm not hoping that we're going to find a magic bug or a magic community that's going to degrade all of the plastic in the ocean. But I do think it provides some clues, especially if we can look for what gets enriched in an association. But yeah, you're absolutely right.